Hi everyone. In this video, we're going to look at the regulation of gene expression in the context of differential gene expression in eukaryotes. Differential gene expression is crucial in multicellular organisms. Since all of the cells were produced via mitosis, they contain the same genetic information. But in order for them to carry out specialized functions, they have to be more selective in which genes are turned on or off. This regulation can be accomplished at multiple stages in the gene expression process, and we're going to zoom in on a few of them in turn. If you consider the process of gene expression as a car driving down a road from one place to another, we can think of the control mechanisms as stoplights down the road. The first stoplight we'll come to is that of chromatin structure. The base unit of chromatin organization is the nucleosome. This is composed of a core of 8 histone proteins, 2 each of 4 types, and the DNA wrapped around the structure twice. The histones have tails that protrude from the nucleosome, and these can be modified by the addition or removal of chemical groups. For example, if acetyl groups are added by enzymes called histone acetyltransferases, or HATs, this causes the chromatin to open up, making it more accessible for transcription. If the acetyl groups are removed by histone deacetylase enzymes, the chromatin closes and transcription is reduced. These are dynamic changes that are frequently altered in the cell, allowing transcription to be fairly rapidly increased or decreased as needed. We're now through our first stoplight and heading towards our second, which is the regulation of transcription. This stage is common to both prokaryotes and eukaryotes. In prokaryotes, much of it is accomplished with the operon system, which we'll review in a separate video. But in eukaryotes, we have a slightly different system of regulation. Now, some genes are transcribed in all cells, and we typically refer to these as housekeeping genes, which are required for general cellular activities, such as those encoding the structural proteins of the cytoskeleton, for example. These genes are under the control of general transcription factors, which are not specific to any individual tissue or cell type. These transcription factors are DNA-binding proteins, which recognize sequences within the promoter region of the gene. Once bound, they can recruit other proteins to assemble the transcription initiation complex, resulting in the binding of RNA polymerase II, which can then begin the process of transcribing the gene. While general transcription factors function in all tissues, some genes are only transcribed in certain tissues. For example, genes encoding proteins necessary for muscle contraction will be transcribed in muscle tissue, but not in the cells responsible for vision in the eye or for filtration in the kidneys. This kind of specificity is accomplished by the binding of proteins called activators to far away or distal control elements, termed enhancers. While there are relatively few activators, they can be grouped in many combinations, allowing precise expression of necessary genes. The activators, if present in that cell type, will bind to the enhancers, and DNA-bending proteins will fold DNA to bring the activators into the proximity of the transcription initiation complex, allowing the recruitment of RNA polymerase and subsequent transcription. Now we have transcribed our gene, we can move on to the next stoplight, which is alternative splicing. We're not going over the details on this process here, but you can review it in the transcription video. Once splicing has taken place, we can move on to the regulation of translation, which, among other mechanisms, can be regulated by a process called RNA interference, or RNAi. Naturally occurring RNA interference results from the expression of non-coding, or NC, RNA molecules, including microRNAs. These are transcribed in the nucleus and form hairpin structures, which are then exported to the cytoplasm. Here, the microRNA is processed, and one of the two strands will be incorporated into a protein complex called an RNA-induced silencing complex, or RISC. The RISC will bind to messenger RNA through complementary base pairing of the microRNA with the 3' untranslated region of the mRNA. And depending on the degree of complementarity, one of two outcomes will occur. If there is a high degree of complementarity between the RNAs, the mRNA can be degraded. More likely, however, is that only a small portion of the microRNA will bind to the mRNA, resulting in the inhibition of translation instead. Together, these processes allow the cell to fine-tune the expression of its genes, more like a dimmer switch than an on-off button. Now our mRNA has been translated, we can move on to the next stoplight of protein processing. In this stage, proteins must be correctly folded in order to be functional, and some will require modifications such as the addition of chemical groups, or may even require portions of the polypeptide chain to be cut out before the protein is completed. This predominantly takes place in the Golgi apparatus, which will then direct the protein to its final destination. Of course, like everything else in a cell, proteins have a finite lifespan, at the end of which they will be degraded. Proteins are marked for degradation by a process called ubiquitination, in which the cell will attach a chain of small molecules called ubiquitin to the protein in question. This targets the protein for degradation by a structure called the proteasome, which then releases the amino acids back into the cell to be recycled into another protein. In this way, the cell can regulate many different processes, including signaling, membrane transport, and progression through the cell cycle, as well as cleaning up proteins that are damaged or no longer needed. The final stop on our tour of regulating gene expression is that of protein transport. In order to function correctly, proteins must be sent to the correct intracellular location. For example, proteins involved in transcription need to be transported into the nucleus, while those involved in facilitated diffusion may be sent to the plasma membrane. And you can review this process in the endomembrane system video in the cell playlist. Thanks for watching, and if you haven't seen it yet, please be sure to check out the video on operons.